Welcome to the deep dive. Today we're taking a really interesting look at BPC157.
hmm
It's a synthetic peptide, very much like a natural protein we actually have in our own stomach juices.
Right, which is kind of fascinating in itself.
It is, and we've basically gone through a lot of the preclinical research here, the scientific literature. Our mission really is to understand what the research says right now, its potential, but also, its limitations.
And one of the first things that jumps out is just how stable this BPC-157 seems to be.
Right.
Resistant to enzymes, stable in stomach acid.
Which is huge, right? Because that hints at maybe being able to take it orally.
Exactly. Unlike a lot of peptides out there, it opens up possibilities.
Yeah, definitely. And the animal studies, wow, they show some pretty remarkable stuff for healing, musculoskeletal tissues especially.
Yeah, quite striking results there.
Okay, so let's jump into that. What did you see about musculoskeletal repair specifically?
Well, across quite a few studies, mostly in rats, BPC-157 seemed to consistently speed up tendon and ligament healing.
Speed it up and make it stronger?
Yeah, potentially.
Yeah.
We're talking about improvements on biochemical, microscopic levels.
Yeah.
Even biomechanical. Like the healed tendons could handle more force before failing in some tests.
Okay. And in the lab, did
Yeah.
it affect the cells directly?
It seems so. They saw accelerated growth of tendon tissue samples, explants, and also quicker migration of the fibroblasts, the repair cells, possibly working through something called the FAK-Paxillin pathway.
Interesting. So it's signaling, get moving, repair this.
Looks like it, yeah. And similar positive results were seen for ligament healing too, not just tendons.
Right. And wasn't there something about muscle attaching to bone? That seems tricky to heal.
There was a study, again in rats, where they detached the quadriceps muscle, and giving BPC-157 early actually helped reverse healing failure. It led to successful reattachment.
Orally, wow. That's stability factor again.
Plus, other animal models showed maybe less muscle scarring and, importantly, better blood flow angiogenesis to the injured spots.
Okay, so a broad healing potential there. Now, going back to its origins, the stomach, what about gut protection?
Well, given it's naturally found and stable in gastric juice, it makes sense it'd be involved in protecting the gut lining. Robert's cytoprotection, they call it.
So protecting the cells directly.
Yeah. And preclinical work showed it was effective against lesions like ulcers or damage in the stomach, esophagus, intestines, even liver and pancreas in some studies.
And could it help against common culprits like alcohol or NSAIDs?
The studies suggested it could counteract the damaging effects of both. Yeah.
That's pretty significant. Did it show potential beyond just damage control in the gut?
Yes. Some animal studies pointed towards preventing or even reversing conditions like adjuvant arthritis, which is an inflammatory model, and also helping heal tricky things like intestinal anastomoses, where bits of intestine are joined, or fistulas.
And wasn't there some very early human research there?
There were some initial clinical trials looking into it for ulcerative colitis. Still early days, though.
Okay, from gut healing to brain effects, that seems like quite a jump.
It does, but the preclinical work suggests connections. Beneficial effects were seen in animal models for things like depression.
Really?
Yeah, and also counteracting some stroke effects in rats, helping resolve neuronal damage, improving memory and motor function afterwards.
So potential neuroprotection and maybe even regeneration.
There's some evidence suggesting it could aid nerve regeneration and reduce neuronal damage after, say, stroke or spinal cord injuries in animal models. It might even help balance neurotransmitters like dopamine and serotonin.
Fascinating range. Okay, what about the cardiovascular system? Heart, blood vessels?
Again, preclinical studies showed promise, counteracting heart failure symptoms, arrhythmias, even thrombosis or blood clots in animal models.
How? What was the mechanism there?
A really interesting finding was its ability to rapidly activate collateral blood flow pathways, sort of like opening up detours when the main road is blocked.
That could be huge in things like blockages.
Potentially, yes. Reversing vascular or even multi-organ failure in some specific animal emergency models. They also saw increased angiogenesis, new blood vessel growth, and linked it to VEGFR2 expression.
VEGFR2, that's a key growth factor receptor.
Yeah, exactly. And it seemed to counteract specific conditions like Budd-Chiari syndrome in rats and rapidly reduce blood pressure issues, whether too high or too low, and blood clot formation.
It really seems to touch on many different biological processes. Is there a common thread? How might it be doing all this?
A lot seems to point towards its interaction with the nitric oxide, or NO, system, especially enos-derived NO. That seems to be a primary target involved in signaling for protection and repair.
Okay, the NO system. Anything else?
Well, that proangiogenic effect, stimulating VEGFR2, is key. It might also nudge other growth factors and modulate inflammatory pathways, generally reducing inflammation signs in animals, and as we mentioned, influencing neurotransmitter systems too.
So it sounds almost too good to be true. Let's get to the crucial part. Safety and, the reality check.
Right? So in animal studies, the safety profile generally looked pretty good. Some injection site irritation, but they couldn't even determine a toxic dose in rats or dogs in some studies. However,
Is there but?
there's the significant but. That proangiogenic effect, promoting blood vessel growth, raises concerns about cancer. Cancer needs blood vessels to grow.
Ah, right. Because things like EGFR signaling, the NO system, even EGR1, which BPC might influence, they can all play roles in tumor growth.
Exactly. So because of that potential risk, it's strongly advised against for anyone with diagnosed or even suspected cancer. That's a really critical point.
Definitely. And what about actual human trials and regulatory approval?
That's the main limitation right now. Human clinical data is very scarce. Mostly phase 1 safety studies, some small pilot studies for things like ulcerative colitis, MS, knee pain, but nothing large-scale proving efficacy and safety definitively.
So no green light from the big agencies.
No. Neither the FDA nor the EMA have approved it. There just isn't enough rigorous clinical evidence yet. And it's worth noting, it's on the World Anti-Doping Agency's prohibited list.
Right. W ought to ban it. And the FDA.
The FDA has also issued warnings about compounded drugs containing BPC157, basically highlighting the lack of approval and safety data.
So we're left with this picture of, well, enormous potential seen in labs and animal models.
Across so many different systems, musculoskeletal, gut, brain, cardiovascular.
But a major gap when it comes to solid human evidence.
Precisely. The preclinical work is compelling, no doubt. But without large-scale, well-controlled human trials, we can't confirm if these benefits translate or if they're safe enough for widespread use. That research is absolutely essential.
It really leaves you thinking, doesn't it? And it's a big if future human trials do confirm these widespread benefits and find ways to manage the risks like the cancer concern, what could that mean for treating injuries, gut issues, maybe even neurological conditions? And maybe more broadly, what other questions does this kind of research raise for you about how we develop and regulate these powerful peptide molecules? Lots to chew on there. Thanks for this deep dive.